Good afternoon to one and all. I'm going to talk about the seminar on the topic characterization of indoor and gut microbial communities in connection with the respiratory health. So this is the overall summary of my presentation. First, I will explain my PhD thesis on this topic, and then I will explain the technology that I'm using in this topic, the next generation sequencing, and then I will explain the two studies, study 1 and study 2, which is concerned with this topic. So, in my PhD topic, we are planning to employ the next generation sequencing and advanced bioinformatics tools to see the microbial communities present in our samples. And the hypothesis in our study 1, which is school intervention studies, is there any qualities which is connecting the moisture damage and the health effect, like microbial communities. And in the study 2, we try to see is there any microbial communities which is relevant to the risk of developing asthma in the children. So this is the overall thing that I'm going to do in my PhD thesis. And this is the overall representation, schematic representation of what I am going to do in this topic. So basically, I'm going to deal with the indoor samples, indoor air sample, and also with the early infant gut microbiome. And we are going to extract the samples from those environments, the DNA, and we are going to use the next generation sequencing platforms with those DNA marker gene based studies, and once we get the DNA, we will process it in the bioinformatics pipeline that we developed in THL, and that will explain whether is there any specific qualities that link the indoor microbiome with the health thing, and also explain whether the early gut microbiome has some effect on developing the risk for asthma. So, in the next generation sequencing, it's kind of very broad technology, the latest technology, in which in our studies we are only using the marker gene based approach, which is kind of a signature for a microbe. So it's kind of a barcode for a microbe that we will identify the microbe in the samples. So these are all the regions. The 16S is for the bacteria in the samples, and ITS is for the fungal identification. So we use those marker genes in this latest technology, and we will. This is the workflow of the whole project. It's like we will extract the DNA and we make the DNA libraries out of it, which is suitable for that next generation sequencing platform, which is Illumina MySeq. From those libraries, we will get the sequencing data which is focused towards this marker genes. So from that data, we will analyze by a custom-made bioinformatics softwares. By analyzing those data, we will came to know that which are all the microbes present in those samples, and we will able to connect with the metadata, and we will try to answer our research questions. So this is the slide for explaining the background of my first study. So we know that if there is a moisture damage in the school buildings, then we know that there is some health effects in the children, and that they develop some some respiratory symptoms and also allergy and those kind of health effects or outcomes, epidemiology based outcomes were seen in the children. So we know that if moisture damage exposure in the children, if there is moisture damage exposure in the schools, then we know that the children are developing symptoms. But the thing is that we don't know what is the mechanism behind it, like why it is causing some health effect in the children. We don't know, and also what is causing those things. We don't know. So our hypothesis in this study is we think there is some microbial communities like fungal or bacteria. There is some microbial communities present in these environmental that cause this health effect. That is our hypothesis in our study. So we made an experimental design that we have taken the moisture damaged school buildings like 11, and we also have included like 5 non-moisture damaged like healthy buildings in our study, and we tried to see whether there is some differences in the microbial community that explain the health outcomes. So that is the main goal of our project. So for that what we did is we collected a lot of health related information from those buildings, like what is the age of the children, and then what is those kind of how long they exposed to that environment, like that, those health kind of information in the metadata. And also we collected the dust sampling and like using the electrostatic dust collector. So we also collect the samples from the environmental, and also we collect the metadata in the studies. So then we try to explain with our aims like how it is connecting with the sequencing data. So this is the status of the current project, that we have finalized these 200 dust samples that collected from the study, like 11 school buildings which is moisture damage and 5 which is non-moisture damaged. And also we extracted the DNA, and it is like about to sequence now, so it will be available. The analysis will be done in the early spring of next year. Then there is a second study which is from the Pasteur consortium, where there is like a 5 so the previous study was only from the Finnish cohort. So in this study it's kind of different project, where we included like a 5 countries like Finland, Germany, Austria, Switzerland and France. So these five countries are included in it's kind of multi-center studies, where we collect the gut microbiome of the early infants, and where we have like a continuous sampling, and then we also measure whether the children are developing asthma, those symptoms. The asthma assessment was done for those children. So here the hypothesis is like there is like a knowledge that the early gut microbiome of the infants had an effect on the immune development. So our hypothesis here is like the microbial community, whether is there any factors which is influencing the microbiome of the gut microbiome, which will develop the risk for asthma. That's our hypothesis. So we are trying to see the microbial composition of the collected gut samples from the infants. So this is the current status of that project, where the sample size is huge, like 1900, to make sure that we have a like a robust finding. And also that one was the DNA sequencing and library preparation was done, and for 16S ribosomal RNA, those data, the bioinformatics are done, but still the main outcome has to be, the more statistical work has to be done. But for the ITS, it is almost going to be done. So that in early spring we will done the analysis for the ITS too, and it is estimated that we can able to make a report in this time point. So I'm very grateful that I got this fund for my PhD from this Kopio Sudan and Sato and Kertu Ya Kale Vikin Rahastu. And also we thank the Academy of Finland for providing money for the project money, and also Theo Serbia Rasto, and also I got fund from the NE and Antiviguri Rasto for my PhD. So I also want to thank everyone who supported my PhD research, and also I want to thank the THL and UEF for providing the platform for do this research, and also my supervisors who are like a backbone for my research. Thank you very much. Thank you. Kiitos.